Okay. Good morning, students. Last class I have discussed about bacterial reverse mutation test, or simply it is called as Ames test. Okay, we are in discussion about genotoxicity studies. So, alteration of the gene structure is called as mutation. The agent which one leads to the alteration of the gene structure, that agent is called as mutagen. To test whether our test chemical can induce the mutation at the level of the gene or not, that bacterial reverse mutation test or Ames test is the predominant or test can be used. Coming to the test for chromosomal aberrations. So, if chromosomes are damaged by the our test chemical or our new chemical entity, to check whether our test chemical or new chemical entity can damage the chromosomes or not, we need to use the test. The test name is called as chromosomal aberration test. So there are basically two types of endpoints. There are two types of endpoints that can be used to determine if a substance can cause chromosomal damage or aneuploidy. So structure of the damage of the chromosome, that is the one endpoint, or change in the number of the chromosomes, that is called as aneuploidy. Remember, if there is a change in the number of the chromosomes, that is called as aneuploidy. If there is a change in the structure of the chromosome or damage in the structure of the chromosome, that is called as chromosomal damage. So chromosomal aberrations and micronuclei, there are two: chromosomal aberrations and micronuclei. The name indicates micro, small nuclei. Both can be visualized under a microscope. We need to visualize it under the microscope. Most chromosomal aberrations are not viable. Most chromosomal aberrations are not viable when, for example, the deficiency comprises essential genes. There is a deficiency, the deficiency comprises essential genes, and thus they are not transmitted to daughter cells. They are not transmitted to daughter cells. When there is a deficiency comprises the essential genes, they are not transmitted to daughter cells. Although micronuclear visualized in cells following the first cell division, after first cell division there is a visualization of the micronuclei. These are not retained in all subsequent generations. That appearance of the micronuclei in daughter cells will be observed only on first cell division. In subsequent generations, there is no observation of the micronuclei under microscope. Based on many genetic studies of the chromosomal basis of heritable genetic effects in humans and other aspects, it can be assumed that compounds able to induce chromosomal aberrations and micronuclei in those states are also able to induce transmissible chromosomal mutations. Transmissible chromosomal mutations, that is, transmissible chromosomal mutations, nothing but reciprocal translocations, stable translocations, and aneuploidy. So location means changing of the amount of the chromosome from one place to another place, that is called as translocation. Reciprocal translocations means opposite. And aneuploidy, you know very well, aneuploidy means change in the number of the chromosomes. Change in the number of the chromosomes is called as aneuploidy. Coming to the test, in vitro mammalian chromosomal aberration test. You are using the in vitro, so we need to take the cells, that is cell culture or artificial cells. The in vitro chromosomal aberration test identify the substances that induce structural aberrations, that induces the changes in the structure of the chromosomes, that is maybe deletions and rearrangements in cultures of the established cell lines. In cultures of the established cell lines, that is example you can take CHO, V79, Chinese hamster lung, TK6 or primary cell cultures including human or other mammalian peripheral blood lymphocytes. Here we are taking the mammalian cell cultures or human cell cultures. The cell cultures measure the cells peripheral blood lymphocytes. Here we are taking the cells peripheral blood lymphocytes. Structural chromosomal aberrations may be of two types: structural chromosome or chromatid. So changing in the structure of the chromosome or damage in the structure of the chromosome at the level of the chromosome or at the level of the chromatid. If it is damaged at the level of the chromosome, that is one thing. If it is damaged at the level of the chromatid, that is one thing. Maybe at the level of the chromosome or maybe at the level of the chromatid. Maybe at the level of the chromosome or maybe at the level of the chromatid, depending on the mechanism of action. Depending on the mechanism of action. So depending on the mechanism of action means our test chemical is damaging either at level of the chromosome or at the level of the chromatid is depending on the mechanism of our test chemical. Okay, depending on the mechanism of action, the chromatid type is most oftenly observed. So the chromatid type is most often observed. The chromatid type is most often observed. Most chromosomal aberrations are observed. Most chromosomal aberrations. Aberrations means changes or damage are observed only in metaphase of the first or second mitotic cell division. So you may observe the most chromosomal aberrations only in metaphase of the first or second mitotic division, because cells containing these aberrations are lost in subsequent cell divisions. So you may get the damaging of the chromosome on first division or second mitotic given, and you can observe in cell division in metaphase. At which phase of the cell division we may observe the chromosomal aberrations? Metaphase. These damages will not transfer to the subsequent cell divisions. You may get observation only or first cell division or second mitotic cell division. Individual cells are viewed by microscope. Yes, we need to observe under microscope, and the information on the types of chromosomal aberrations seen in the cell is recorded. That will be recorded. Chromosome mutations occur if DNA strands breaks or misrepaired by non-homologous end joining. This point I don't want to describe it here. Here also. Coming to the next, second test is in vitro mammalian cell micronucleus test. Second test is in vitro mammalian cell micronucleus test. The in vitro micronucleus test identifies the substances that induce chromosomal breaks and aneuploidy. So the in vitro micronucleus test identifies the substances. If this test can identify the substances that induce chromosomal breaks and aneuploidy, micronuclei are formed. When there is a chances of the formation of the micronuclei, micronuclei are formed when either a chromosome fragment or an intact chromosome is unable to migrate to a mitotic fold during the anaphase of the cell division. Generally, at anaphase, the chromosome need to move to the opposite pole to make the daughter nuclei. So cell division is two types, that is nuclear division, cytokinesis. Nuclear division means division of the nucleus, division of the nucleus into two nucleuses, and cytokinesis means transfer of the cellular contents equally to both daughter cells. Here the micronuclei are formed when either a chromosome fragment or an intact chromosome is unable to migrate to a mitotic fold during the anaphase stage of cell division and is left out of the daughter nuclei It come back to it is not transferred to the daughter nuclei, left out of the daughter nuclei. The test thus detects both structural chromosomal breaks caused by clastogens. The agents which are break the chromosomes, that agents are called as clastogens, or numerical chromosomal abnormalities. Numerical means number. Numerical chromosomal abnormalities or chromosome loss caused by aneuploidies. Aneuploidies can bring the changes in the number of the chromosomes. Clastogens can make the breaking of the chromosomes. chromosomes. The agents which can make the breaking of the chromosomes that are called as clastogens, the agents which can change the chromosome number or chromosome loss is called as anogens. Here the use of FISH, fluorescent in situ hybridization, fluorescence in situ hybridization. The fluorescence in situ hybridization and centromere staining, we can use the staining to the centromere, can provide additional mechanistic information and can help in differentiating clastogens micronuclei without centromeres. Our clastogens can make the micronuclei without centromeres from anogens. Anogens can make micronuclei with centromeres. If our substance is the anogen, means it can change the chromosome number and it can change the chromosomal loss. So that anogen leads to the micronuclei with centromere. Clastogen leads to the micronuclei without centromere. For example, if our substance is the clastogen, what is the end point in case of micronuclei without centromere? Micronuclei without centromere, whereas anogen means it can make the micronuclei with centromere. The test can be conducted using cultured primary human or other mammalian peripheral blood lymphocytes. Here we are taking the cells is either human or mammalian peripheral blood lymphocytes, and a number of cell lines such as already I told CHO, V79, CHU bar, slash IU, L5178Y and TK6. There are other cell lines that have been used for the micronucleus assays H229, CaCO2, HepRG, HepG2 and primary Syrian hamster embryo cells, but these have not been extensively validated, and the test guideline recommends that they be used only if they can be demonstrated to perform according to the described requirements. The scoring of micronuclei is generally conducted in the first division daughter cells. Cytochalacin B can be used to black. So this is the, remember, very important, cytochalacin B can be used to black cytoplasm division, nothing but, or cytokinesis, and generate binucleate cells. Binucleate cells means two nucleate cells during or after test substance exposure. If the cell is undergoing the exposure to the test substance, okay, we need to use the cytochalacin B. This may be desirable because it can be used to measure cell proliferate and allows the scoring of micronuclei in dividing cells only. We need to observe the micronuclei. The use of cytochalacin B is mandatory for mixed cell cultures such as whole blood cultures in order to identify the dividing target cell population. But for cell lines, the test can be conducted either with or without cytochalacin B. The scoring of micronuclei has potential for automation, flow cytometry or image analysis. Automated systems that can measure micronucleated cell frequencies include but are not limited to flow cytometers, image analysis platforms, and laser scanning cytometers. So this is the scoring of the micronuclei. That are the techniques we need to use the scoring for the micronuclei. The in vitro micronuclei test has been shown to be as sensitive as the chromosomal aberration test for the detection of clastogens. So for the detection of the clastogens, we need to use the in vitro micronucleus test, and has the additional advantage of detecting anugenic substances. As anugenic substances can be classified, can be observed by in vitro mammalian cell micronucleus test. Okay. However, the in vitro micronucleus test detects only chromosomal fragments. The visible chromosomal aberration test detects translocations and other complex chromosomal rearrangements that may provide additional mechanistic information. So here totally I have discussed three tests. The three tests are: one is in vitro bacterial reverse mutation test, which is also called as Ames test. Second one is, that is, chromosomal aberration test. In chromosomal aberration test, okay, changes in the structure of the in the structure of the chromosome, okay, or that is number of the chromosome changes, that is aneuploidy. Third one is I have described the final one is in vitro micronucleus test. These three steps are used to classify the substance whether it induces the changes in the structure of the DNA or whether it induces the changes in the structure of the chromosome. Okay. So thank you. If you have any doubts, you can ask.